Hello and welcome to today's lecture of Introduction to Mechanobiology. In the last class, we had started discussing about the nucleus, which is the centerpiece of the cell and which serves as the signaling point where gene expression is turned on during processes like differentiation. So, we had started discussing about the structure of the nucleus, and in this context we had introduced you have proteins or lamins which are intermediate filament proteins, which serve, and these lamins are part of the LINC complex. LINC stands for linker of nucleoskeleton and cytoskeleton. So, in the nucleus has two membranes, inner nuclear membrane and outer nuclear membrane. Underneath the inner nuclear membrane, you have these networks of two lamins, lamin networks, lamin AC and lamin B. B can be either B1 or B2, and AC is because of splice variant of the lamin A gene. And outside, you have the cytoskeletal network through which you have connections through the nesprins. So, this establishes the direct connection between outside the nucleus and inside the nucleus. So, lamins not only bind to the heterochromatin, there are various nuclear envelope proteins or NEPs, for example, emerin, sun proteins, etc. These participate in this cross signaling, and the LINC complex serves as a force transducer. So, force might lead to protein unfolding of various proteins within this network, might lead to reinforcement of lamina, or lead to chromatin stretching. Now, also discussed this paper last class where we showed that lamin A to B ratio varies depending on tissue elasticity. So, this lamin A to B ratio roughly has been shown to scale as e to the power 0.6, where E is the elasticity of the tissue. So, for example, in mesenchymal stem cells, they have high lamin A to B ratio. In muscle cells, muscle or heart or lung, you have moderate levels of lamin A to B ratio, while in brain cells, neuronal cells, you have low. And here when I say low, there is A to B ratio is less than 1. The other aspect which I introduced in last class was that lamin phosphorylation scales inversely with stiffness. In other words, on cells cultured on stiff surfaces, phosphorylation is less. So, phosphorylation of serine 22 is less on stiff surfaces, but is high on soft matrices. So, if you look at the nuclei sizes inside the cell, if you think of an embryonic stem cell, the nucleus occupies nearly 90% of the whole volume. while in a differentiated cell like a fibroblast, this is order 10 to 15%. Okay. You can clearly see. So, for embryonic stem cell, this is what the nucleus will look like. Okay. There is very little or no cytoskeletal network. So, you just have the nucleus occupying the gamut of the space, versus for a fibroblast the nucleus is right at the center and it takes a much less amount of space, and it is also more elongated under a lot more stress, because you have cytoskeletal fibers which actually push down on the nucleus. In this regard, it has been shown that there are perinuclear actin caps which push the nucleus down. Okay. So, in terms of deformability of the nucleus, okay, one would imagine that deformability, nuclear deformability, will be high in stem cells. Okay. And why is that? So, in an embryo, if you talk about an embryo, okay, a deformable nuclei could contribute, okay, could actually migrate much faster or squeeze through much faster. Okay. So, to do this, to assay how nuclear deformability varies between stem cells and differentiated cells, a simple experimental setup that has been used is the experimental setup of micropipette aspiration. Okay. So, what you do is you have a cell, okay, if this is your cell with the nucleus, you put a pipette, a micropipette, okay, and you expose this cell to a step jump in pressure. Okay. This is a pressure difference. So, this is equivalent of sucking on a cell. So, what you will have is, as time goes on, the cell and the nucleus, so the cell will have a shape like this, and the nucleus will also deform in this kind of configuration. Okay. So, what you can track is this length of the cell and this length that the nucleus has entered into the micropipette. Okay. So, this is ideally if the nuclei was more deformable. Okay. So, what you should have is, when you suck, just like the cytoplasm will move in, the nucleus will also move in. Now, what the group has tracked this deformability as a function. So, you can track the ratio of Ln by Lc as a function of differentiation. Okay. So, what has been shown is as a function of differentiation. So, initially, okay, so Ln by, and you are plotting Ln by Lc here. So, at early times, so when it was in pluripotent state, this ratio is as high as 0.9, okay, but this drops rapidly as the cell starts to differentiate. Okay. And at large time points, let us say order 8 days, this ratio drops to as little as 0.2. So, this demonstrates that the nucleus stiffens as it differentiates. So, if a nucleus stiffens as it differentiates, what is the link on cell migration? Okay. So, ideally a soft nucleus, okay, should mean that a cell is more invasive. Okay. So, why? Because in 3D matrices, when cells are migrating, the cell has to squeeze through the pores in the matrix. So, if the matrix is very dense, it is very difficult for the nucleus to be squeezed. Okay. So, there has been seminal study by Friedel and Wolf, okay, and then they showed that when a cells migrate through matrices. So, what they did was they tracked cell migration through collagen gels of increasing concentration. Okay. And what they showed that if the cell is secreting MMPs, then whatever be the concentration does not matter, because the cell will secrete MMPs and they will degrade the matrix, thereby making holes which are big enough for the cell to squeeze. However, in the presence of MMP inhibitors, so GM6001 is broad spectrum MMP inhibition. Okay. So, then you have a drop in migration speed. Okay. And for pores, okay, which are smaller than 10% of undeformed nuclear cross section. Okay. So, there is complete migration arrest, meaning that cells are completely stuck. Okay. So, this limit the authors named as the nuclear deformation limit. Okay. So, in other words, a nucleus cannot be deformed to below 10% of its original undeformed cross sectional area. Okay. So, consistent with this, if you think of leukocytes or dendritic cells, their nuclei, okay, do not express Lamin AC. Okay. So, this makes the nuclei more deformable, so that leukocytes can go all over the place within the tissue. Okay. So, this suggests that there is a relationship between Lamin levels, which dictates its nuclear stiffness, and this impacts its migration ability. Okay. So, to test this, so I will discuss this paper. Okay. Okay. So, what these authors did, okay, was took cells, okay, 3 different types of cells, A549, okay, mesenchymal stem cell, and U251. Okay. So, these have different A to B ratio. Okay. So, for A549s, this is 2.3, MSC have high, they have A to B ratio of 8, and U251 have 0.8. Okay. So, A to B ratio is varying in these cells, okay, while B ratio, okay, B1 and B2 are roughly comparable. Okay. So, they asked, okay, so they used transfer migration ports. Okay. What are these? Okay. So, these are transfer migration inserts in which you plate cells on the top, okay, put a gradient, you put a gradient in serum. So, this acts as a chemotactic cube, making the cell squeeze. Okay. But if you reduce, so this is your port size. Okay. So, if the port size is comparable to the nuclear size, so ideally there should be no difference. Okay. But if your port size is other 3 microns, then the real picture is something like this. Okay. This is your cell and this is your nucleus. Okay. It has to deform significantly to make it through the pore. Okay. So, what they counted was they plated the same number of cells on top. So, this is your top of your ports and this is the bottom of your ports. Okay. And then they asked that how many were the number of cells at the bottom. Okay. And what they found was So, U251 cells which have the lowest Lamin A to B ratio. Okay. So, so maximum number of these cells were able to transit, and it was lowest in case of A549 cells. Okay. So, suggesting once again that when Lamin A to B ratio is high, so this is number of cells at the bottom of the ports. Okay. So, in U251 cells you had the maximum efficiency of migration because the A to B ratio was low and these cells were soft. Okay. But what they also observed was when they looked at the nuclei underneath, okay, in the bottom ports, many of the nuclei, okay, they exhibited these blebs. Okay. So, if this is a regular shape of a nuclei, after migrating through the ports, they had these kind of shapes. So, these are examples of nuclear blebs. Okay. And what they found was, in general case, A and B networks, okay, Lamin A and B are juxtaposed. Okay. But in these nuclei, there was a separation or segregation of Lamin A and B. Okay. So, first of all, not only were these cells nuclei were elongated, okay, but you have these odd shaped nuclei and the segregation of Lamin A to B. Okay. So, this segregation of Lamin A to B and all this was only observed in 3 micron ports. It was not observed in 8 micron ports, where the ports are big enough that the nucleus does not need to deform much. Okay. So, this shows that having low Lamin A to B ratio actually helps the nuclei to migrate. Okay. And during the migration, there is significant amount of change in nuclear shape, okay, which correlates with the migration process. Okay. So, what they then did, okay, they compared the nuclear. So, if you have a nuclear shape like this, you can track the shape by using a measure of circularity, okay, which returns a value of 1. Okay. So, it is a value between 0 and 1, 1 means perfect circle. Okay. So, in U251 cells, okay, which had low Lamin A to B ratio, okay, there was no change before and after. Okay. At the top and the bottom, the circularity remained unchanged. Okay. But in case of mesenchymal stem cells, okay, the bottom cells were much more elongated. Okay. And what they also found, okay, was you could track this change in stiffness, okay, with the Lamin A levels. Okay. So, what it also suggested, so based on their experiments, they suggested that Lamin A acts like a viscous damper. Okay. And Lamin B acts as an elastic spring. Okay. In U251 cells, okay, as soon as it deformed, when they let go, when they came underneath, they immediately regained their shape. So, that is why, and they had A to B ratio of 0.8, which is less than 1, which means that Lamin B was significantly high in these cells. Okay. In these cells, there was no change in circularity, but in these cells, in MSCs as well as in A549 cells, okay, which had much greater Lamin A to B ratios, you found a significant drop in this nuclear shape. Okay. So, you have, so mechanically you would represent this network to Lamin B would be the spring and Lamin A would be this dashpot. Okay. So, the Lamin B network, when it deforms, after deformation is removed, because it is an elastic network, so this deformation is reversible, while the Lamin A network, which is a viscous damper, whatever you deform, you do not regain, you lose that. So, you have irreversible deformation. Okay. So, you have two different deformations. What they also observed, that given that there was some heterogeneity in the population, okay, so the ones at the bottom, okay, so they checked. So, you have the top cells and the bottom cells, and then they checked what is the circularity of the cells at the top and the bottom, okay, and they found, okay, but the cells which were at the bottom also expressed lesser amount of Lamin A ratio. It was less compared to those at the top. Okay. So, even for the cells which migrate, so you have Lamin A levels, okay, leads to sorting in 3D migration. Okay. So, you would see that there is a greater amount of Lamin A in the cells which remained at the top or were unable to migrate compared to the cells which did migrate to the bottom. Okay. But there is one cost of low Lamin A levels. What they found was in the cells which exhibited less amount of Lamin A expression, okay, when there was low Lamin A expression, these cells also had a tendency to apoptose after they migrated. Okay. And this, they did this assay by probing the caspase 3, cleaved caspase 3, okay, the proportion of cells which had positive cleaved caspase 3. So, suggesting that while Lamin A is needed, low Lamin A enhances migration, but this migration induced stress, okay, you have migration induced stress leads to apoptosis, okay, suggesting that the Lamin A not only is needed for lower Lamin A is needed for deformability, but lower Lamin A makes the cells more susceptible to cell death. So, Lamin A has a protective role, it protects the nuclear from damage. Okay. And what they also observed, okay, that when they did a deep knockdown of Lamin A, so when they did a deep knockdown of Lamin A using siRNA, okay, what they found was in these cells you have this HSP19 heat shock protein is a protein which is associated with DNA damage, okay, or DNA repair. Okay. Expression of HSP90 was significantly dropped. So, siRNA led to significant drop in HSP90 cells. So, these cells can not only will have more amount of DNA damage, but they lose the ability to rescue themselves. Okay. So, this shows that you have a relationship that lower Lamin A leads to more migratory potential, but it also leads to more death, okay, due to stress. So, essentially you need to optimize the amount of Lamin A so that you are also deformable and you can also migrate and you do not die. Okay. So, consistent with these observations, what has been found is in most of the cancer cells. So, there was a study which compared the expression of these proteins, so LINC proteins in cancer cells, how their expression is altered. And what they found was, say, it compared MCF10A, okay, MCF7 and MDMB 231, and what they found as, so this is normal mammary epithelial cells, okay. This is not non-metastatic and this is highly metastatic. And what they found was, as the cells became more and more metastatic, the LINC protein expression was going down, and these included SUN1, SUN2, Nesprin 2 and Lamin AC. Okay. So, expression of all these proteins was going down. Now, even though this expression might go down, just nuclear softening may not be sufficient for having the cells to migrate. So, you still require, given that the nucleus is so huge, you require additional forces on the nucleus to deform. Okay. So, there was a study which recently demonstrated that myosin 2B is required for nuclear translocation. Okay. And what they showed was when they knocked down 2B, the average nuclear height increased. Okay. So, this was indirect hint that if, as opposed to a nuclei being very thin, if you knock down myosin 2B, if the nucleus becomes big, then it will be that much more difficult to squeeze through a small pore. Okay. And then they subsequently showed that there is, accumulation of, so if a cell is migrating, so they used a variety of pillars, okay, where the spacing was reduced, okay, and they tracked the cell migration against an EGF gradient, okay, and then they showed that whenever the nucleus was trying to pass through these pores, okay, you had an accumulation of myosin 2 at the rear. So, you have 2B at the rear, and what this is doing, 2B is actually squeezing the nucleus. Okay. So, even though nuclear softening helps, you still require myosin generated forces to pull the nucleus forward. Okay. With that, I end here today, and there will be several reading assignments for this class. Okay. Okay. This was a physical limits paper which showed that the nuclei cannot be deformed beyond 10%. This was the paper which I discussed today just now. And this is a recent paper which showed that myosin 2B is required for pulling the nucleus forward. With that, I stop here today. Thank you for your attention.